topic today of my talk is 3D printed oral solid dosage forms for customized drug release. So 3D in pharmaceutics, 3D printing in pharmaceutics has gained considerable interest the last years, the recent years, and I think this is because this is a simple and a single-step process, and also it brings the element of personalized medicine. Of course, already nanomedicines has bring this element, but in 3D printing, it could find its way for special populations like geriatric or pediatric populations, or even in orphan drugs. Also, it has got the advantage that we can prepare formulations with very small amounts of actives. So this is really important and a good advantage over all other processes. Most commonly used 3D printing technologies, and has been already mentioned from the first presenter, are fused deposition modeling, inkjet printing, extrusion printing, stereolithography, selective laser sintering, powder bed printing, and drop-on method. Today I'm going to focus to fused deposition modeling, and in essence the operating principle is the successive deposition of polymer of molten polymeric strands with specific and standard diameter through heated nozzles. So the nozzle is fed with the polymer, and then we have material deposition on a heated bed, usually in a layer by layer mode. Moving to the next slide, we can see the spectrum of the 3D printed formulations and for different route of administrations. So we can see either devices or dosage forms. Oral delivery, either per oral or intraoral, at the moment monopolize the dosage forms. We can see different shapes for different populations, like for pediatric populations. Also ODTs and the buccal films are getting more and more popular. Of course, there are also other routes of administration, like skin delivery with microneedles already in the literature, and also implants for gynecology or wearable devices. So today I'm going to briefly discuss how we can change the release profiles of oral solid dosage forms by changing certain parameters using 3D printing technology. Starting with the first example, when we can see how we can change the release profile by changing the geometry of the dosage form. So in this case, we fabricated with an FDM printer a donut-shaped solid dosage form. Actually, it was comprised of 3 parts: an upper and a lower layer. The upper part and the lower part was from a polylactic acid, which is a biocompatible polymer, and it is non-dissolvable. And the middle layer, in the composition shown in the slide, was polyvinyl alcohol, hydrochlorothiazide as a model drug, and mannitol as a plasticizer. Also, we can add an inner binding ring of PLA, just to be sure that we don't have separation of the PVA compartment. And to the right side of the slide, you can see the cross sections of the filament. It was quite smooth, and also close up of the solid dosage form, when you can see the distinct deposition layers. In some cases also we could see some fusion and some defects, but this is due of course to the limitation of the approach and the method we use. Of course we did all the routine physicochemical characterization to check any crystallinity of the drug and the decomposition, any decomposition of the raw materials and the filament. And we moved to the release studies. In this case, release studies were done in a pH 1.2 in a simulated gastric fluid and also in sieve with a pH 6.8. As a control, we used a marketed product containing hydrochlorothiazide in the same strength like the tablets, the dosage forms we prepared in our lab. So what you can see actually is that in the marketed product, we had a rapidly release almost in 10 minutes, 90 or 10 or 100% of the drug was released in the medium. On the contrary, in the case of the fabricated dosage form, 3D printed dosage forms, we had a linear increase, almost obeying to zero order kinetics with good linear regressions. We complement this study also with with a time-lapsed with time-lapsed X-ray microfocus computer tomography. So in essence, we did exactly the same protocol. We did the dissolution test, but at the different time points, we removed the sample from the medium, and we tested that in the X-ray instrument in order to see what is the mechanism of the dissolution. And what we saw is that we have an even and smooth dissolution of the PVA material from both sides. At the end, of course, we could see a detachment of the upper layer, and most probably that was because of the mechanical stresses once we were removing the tablet from the dissolution medium to check it to the instrument. So also cross sections of the tablet revealed that the printing method was quite successful, although we could see some small gaps, and as I said before, this is because of the limitation of the method. Moving to the second case, we tried to develop pH-responsive 3D printed multi-compartmental dosage forms. So in this case, as you can see, we prepared again solid dosage forms with the upper part from PLA, polylactic acid, and the bottom part was a mixture of polymethacrylates, Eudragid 100 and Eudragid S11. Both of them are pH responsive, are soluble above pH 5.5 and 7 respectively. And then we loaded the compartments of our solid dosage forms with beads of sodium arginate containing the chemotherapeutic agent 5-fluorouracil, and we coated them with chitosan. Chitosan is a known biodegradable material, and also it's known for its bioadhesive properties. So in this case, we tried to deliver the active compound to the colon, which is which is desired in order to avoid off-target toxicity in the epithelium area, and also to reduce the risk from myeloma suppression. So in this case, we developed several formulations as shown in the table, and we ended up with a formulation F3 with the mixture of the polymers and also tech as a plasticizer. Both the other four formulations unfortunately didn't work because the two first we had, because of the fast erosion of the mixture of the polymers, we had fast release, and for the other two, they had very poor erosion, so the release of the active was very slow. Electron microscopy studies revealed that upon coating with the chitosan, the surface of the particles was much smoother compared with plain particles without chitosan. Also, again, we got from the cross sections of the filament quite smooth filament, and at the end, you can see at the bottom, to the right part, you can see that the adhesion between the two layers was quite good. Again, we performed the release studies in two different media, in SGF with a pH 1.2, then in 6.8 using simulated intestinal fluids, and at the end, pH 7.4, trying to mimic more or less the upper and the lower intestine. So what we noticed was that the release of the active in SGF and in SIF was almost negligible. However, at pH 7.4, we could see a rapid increase a release of the active in the medium for all three formulations. Of course, in the case of the formulation that was not coated with chitosan, the release was much higher, and this could be attributed compared to the other two, and this is could be attributed to the interactions between the amine groups of chitosan and the carboxyl groups of sodium alginate. So that was the reason that in the other two formulations, the retardation of and the diffusion of the API was slower, resulting in these two graphs. We did exactly the same study again with microfocus with X-ray tomography, and this time more or less we followed the same protocol. So the first scan was in the native, in the dry form for our formulations. Then we immersed in the medium for 2 hours our dosage forms in SGF, followed other 2 hours in the SIF, and we ended with 5 hours in PBS. And actually what we saw was that in SGF, more or less, the particles were intact, the alginate beads were intact, and only when they were immersed in the SIF medium, we could see some changes, a kind of distortion of the particles. However, there was a dramatic difference in the PBS when a large number of the particles was already escaped from this mess of the polymers, and actually what we could see was that we had the layers and not particles anymore. And if we'd like to sum up with the whole phenomenon, we could see it to the right, to these two images, when we can see at the beginning how was the bottom part of our formulation, and how was at the end. You can see that there is no barrier anymore. This mixture of hydrogids have been already disappeared. The next case I would like to present is 3D printed solid dosage forms for combination therapy. So in this case, we chose two actives, glimepiride and metformin. Both of them are for the treatment of type 2 diabetes. And the main task, and it was quite challenging, was to co-formulate them in the same dosage form. The main difficulty is that they have different dose regimen. That means that glimepiride is once per day, and metformin is 3 or 4 times daily. And also the daily dosing is different by the 2 orders of magnitude. In the case of glimepiride, it's only the dose range between 1 to 4 milligrams, and in the case of metformin, between 500 up to 1,000 milligrams. So in this case, again, we used two dual extrusion this time with the compositions shown in the slide. Again, PVA, mannitol, calcium stearate for glimepiride, and PLA, eudragit, and PEG for metformin. And here you can see in the CAD the location of the drugs in the solid dosage form. Again, we got some electron microscopy results. In the left image, you can see how was the dosage form using a single screw extruder, and in the right part you can see that we have fusion using a twin screw extruder. Again, we used X-ray tomography, but this time we wanted to see the deviation of the printed objects compared to the nominal values of the CAD design. So as you can see, we have the scale and moving from blue to red, we have large deviations of the printed object compared to the nominal values. Actually, there were spots of orange and and red colors only at the edges of the particles. So that means that we had over deposition of the material, and that was mainly because there was some time to switch from one nozzle with the filament to the other. And again, with these compositions, when we did the release studies, as you can see, we had the fast release within 2 hours of glimepiride, which is something we would like to have, and also prolonged the release of metformin. There was a small difference between the two when we did the formulation, the filament with the two different extruders, but this difference was not statistical different. And at the last example, we tried to formulate some osmotic 3D printed dosage forms. If we'll go a little bit back to remember how an osmotic pump is working, we have the semi-permeable membrane, and once the solid dosage forms will be in the intestinal environment in the gastrointestinal tract, then the water from the gastrointestinal fluids will be absorbed through the membrane via the osmosis, and then they will create an osmotic pressure, and the drug in the core through the small holes will be released in the intestinal tract. So more or less we followed the same pattern, but with some modifications. We did 3 different types of solid dosage forms. One, of course, it had the the semi-permeable membrane from cellulose acetate. The core was PVA, was also dealt as a model drug, and as osmogens we had sodium chloride and mannitol. And then we had two other types of dosage forms. Of course, we had the typical small hole on the top of the dosage form, and then we have linear cavities just to help the fluids and the water to penetrate the membrane, so to induce the pressure in the core. However, in our approach, we had two small modifications with the design. And you can see it here in the figure. The tablet could open. It wasn't like an oyster like dosage form. So it could open in the first case when we had the C tablet only from the top, and when, the B tablet only from the top, and when we had the C form, it could open from both sides. We did again release studies, and what we obtained from the release studies was that for form A, which was the tablet that could open from both sides, and form C, which was the tablet with no cover on the top, more or less we had a fast release during the time scale of 200 minutes, and then we had a steady state. The release profile was a little bit different in the case of the tablet with only one opening from the top. Again, we did some X-ray tomography studies, and here we present in this graph the release behavior of the tablet with both openings. So in row A, we have at different time points the way that the tablet behaves in the dissolution medium. And in row B, you can see also the bright spots. The bright spots were the sodium chlorides, mainly were the osmogens, and also this could be verified by elemental analysis in the dosage forms. In figure C, we nicely presented them with colors all this process from the time that the water penetrates or is absorbed through the membrane in the core until the end of the experiment. So if we would like to see in the graph to the left side, we can see that with the green light, which is the osmogen, over the time it is decreased, and actually we can see it here as the green color disappear, and the blue color, which is when the core is getting wet and water or the medium is already in the core, we can see that it covers all this area. And at the same time, at the end of the experiment, we have the detachment of these two layers. The total core volume, depicted with the yellow line, was more or less almost the same, and also we didn't notice big changes to the cellulose stearate core over the experiment. So if I would like to summarize based on the results I have presented so far, we have the ability to host multiple active ingredients resulting in different release profiles, the ability to construct complex formulations resulting in desirable characteristics, and also the ability to arrange excipients and the drug-free areas, resulting in modifying release. I would like to thank my colleague, my former PhD student, Dr. Christos Dimitrios. In briefly, I presented his thesis in this short talk, and also my colleagues, Dr. Kambatsamenis from University of Southampton, for performing the X-ray studies, and the other two colleagues, Dr. Zedis and Dr. Bouropoulos. For all these studies, we have done a lot of mechanical tests, but there was no time to present all this, and these colleagues were responsible for these tests. Thank you very much for your attention.